by talking about eukaryotic pathogens. These include fungi, protozoa, and helminths. They're called eukaryotic because their cells have internal structures, like a nucleus, mitochondria, and endoplasmic reticulum, all surrounded by membranes. This makes them more similar to human cells than bacteria or viruses. And that's important. Why? Because it means it's harder to treat infections caused by these organisms. If we use drugs that target them, there's a higher risk of harming human cells too. Out of these three, fungi are usually easier to grow in lab settings, but protozoa and helminths are harder to culture. So we rely more on microscopy and symptoms to diagnose infections caused by them. Section 2, fungi. Fungi can be unicellular like yeasts or multicellular like molds. They are heterotrophs, which means they don't make their own food. Instead, they absorb nutrients from their surroundings. But unlike animals, fungi absorb nutrients by first releasing digestive enzymes into their environment. These enzymes break down the food outside their bodies, and then the fungi absorb the digested material. Fungal cell walls are made of chitin, a tough and flexible substance also found in the shells of insects. This is different from bacteria, which have peptidoglycan, and plants, which use cellulose in their cell walls. Fungal structures and types. The body of a fungus is called the thallus. In yeasts, the thallus is just one single cell, but in molds, it includes thread-like structures called hyphae. These hyphae can be septate, divided into segments by walls, or choanocytic, meaning no walls at all, just a long cell with many nuclei. Then we have dimorphic fungi. These fungi can switch between yeast and mold forms depending on the environment, like changes in temperature. This switching ability is common in human pathogens, and it makes them harder to diagnose. Fungal reproduction. Fungi reproduce asexually using spores. In molds, you'll see sporangiospores made inside a sac called a sporangium, sitting on a stalk, and conidiospores, or conidia, not enclosed in a sac and produced on structures called conidiophores. Yeasts don't use spores. Instead, they reproduce by budding. A small daughter cell forms off the parent cell. Fungal infections, mycoses. Fungal infections are called mycoses. These can be allergies triggered by fungal spores, opportunistic infections. These only affect people with weak immune systems, like AIDS patients or those on chemotherapy. And true pathogens. These are rare but dangerous. They can infect healthy people by actively invading and damaging tissues. Section summary. Fungi are eukaryotic, heterotrophic organisms with cell walls made of chitin. They reproduce using spores or budding. They may exist as yeasts, molds, or switch forms, dimorphic. Fungal infections, mycoses, range from allergies to serious diseases. Review questions. What makes fungi different from bacteria in terms of their cell wall? Fungi have chitin, while bacteria have peptidoglycan. 2. What is the difference between septate and choanocytic hyphae? Septate hyphae have cross walls, choanocytic hyphae do not. 3. How do yeasts reproduce? By budding. 4. What is a dimorphic fungus? A fungus that can switch between yeast and mold forms. Would you like me to continue with protozoa and helminths next? Let me know and I'll write part 2 in the same slow, easy-to-listen format. Great! Here's part 2 of your Lab Exam 2 audio lecture, covering protozoa and helminths in the same protozoa. Protozoa are microscopic, unicellular eukaryotes. That means they're made of one cell, and that cell has a nucleus. But here's something important. Protozoa do not have cell walls. This makes them flexible, and many of them can move. They're usually found in wet environments, like ponds, soil, or inside the human body. Some live freely in nature, but others are parasitic, meaning they live off a host, sometimes causing disease. All protozoa have 3 main traits. They are unicellular, they are eukaryotic, and they are wall-less. Life stages. Trophozoite and cyst protozoa often exist in 2 forms. 1. Trophozoite. This is the active, feeding, and motile form. It's found when conditions are good. 2. Cyst. This is the dormant, protective form. It allows the protozoan to survive tough conditions, like dryness, stomach acid, or disinfectants. This cyst trophozoite cycle is important for transmission. For example, drinking water with cysts can make you sick. Once inside the body, the cysts turn into active trophozoites. Movement structures. Protozoa move in different ways, but only in the trophozoite stage when they're active. There are three main methods of movement. 1. Cilia, short hair-like structures that help them move, e.g. Balantidium. 2. Flagella, long whip-like tails, e.g. Giardia. 3. Pseudopodia, false feet made by streaming the cell's cytoplasm, e.g. amoeba. There's also a special group called apicomplexans. They don't move on their own. Instead, they rely on hosts or vectors, like mosquitoes. Example, Plasmodium, which causes malaria. Section 4, Helminths. General characteristics. Now let's talk about helminths, also known as parasitic worms. Helminths are multicellular and eukaryotic, just like animals. They don't have cell walls, and they often live inside their hosts, taking nutrients or damaging tissues. Most helminths are parasites, and they are much bigger than bacteria or protozoa. Three main groups of helminths. There are three main types of parasitic worms. 1. Cestodes, tapeworms. These are long, flat, and segmented. They don't have a digestive system. They absorb nutrients through their surface. The head is called the scolex, which has hooks and suckers to attach to the intestine. The segments are called proglottids, and they contain eggs. Tapeworms are covered in a tough outer layer called a cuticle. Their eggs and larvae can infect other hosts. 2. Trematodes. Flukes. These are flat, non-segmented, and leaf-shaped. They have a mouth, but only a partial digestive tract, no anus. They use oral and ventral suckers to attach to organs. They're named after the organs they infect, like liver fluke or blood fluke. 3. Nematodes. Roundworms. These worms are cylindrical and unsegmented. They have a complete digestive tract with both a mouth and anus. Some are free-living, but many are parasites. Examples include Ascaris and hookworms. Which are most similar to humans? Out of fungi, protozoa, and helminths, the organisms that are most similar to humans are helminths. Why? Because they are multicellular animals, just like us. That's also why treating helminth infections is tricky. Many drugs that kill worms can also harm human tissues, because our cells work in similar ways. Section summary. Protozoa are unicellular, wall-less eukaryotes. They can switch between active trophozoite and dormant cyst stages. They move using cilia, flagella, or pseudopodia. Apicomplexans don't move on their own. Helminths are large, multicellular parasites. Cestodes are tapeworms, flat, segmented, no digestive tract. Trematodes are flukes, flat, leaf-like, partial digestive tract. Nematodes are roundworms, cylindrical, complete digestive tract. Helminths are the most human-like of the pathogens, which makes them harder to treat. Review questions. 1. What are the two main life stages of protozoa? Trophozoite, active, and cyst, dormant. 2. Which protozoan group doesn't move on its own? Apicomplexans. 3. What's the function of a tapeworm's scolex? It attaches to the host's intestines using hooks and suckers. 4. Which helminth has a complete digestive tract? Nematodes. Roundworms. 5. Why is it hard to treat helminth infections? Because helminths are similar to humans, so drugs might harm human cells too. Let me know if you'd like part 3, where we cover Enterobacteriaceae, coliforms, and selective media in the same detailed and natural voice format. Great! Here's part 3 of your Lab Exam 2 audio lecture, focused on Enterobacteriaceae, coliforms, and bacterial media and tests. 5. Enterobacteriaceae, the enterics. Now let's focus on a major group of bacteria called the Enterobacteriaceae, or just enterics. These are gram-negative, rod-shaped, and facultative anaerobes. That means they stain pink in a gram stain, are shaped like rods, and can grow with or without oxygen. Most enterics live in the intestinal tract of humans or animals. Some are normal flora and others are pathogens that cause disease. They're incredibly important in medicine because they can cause urinary tract infections, sepsis, and intestinal diseases. What is a coliform? A coliform is a special subgroup of enterics. These bacteria ferment lactose, produce acid and gas, are non-spore-forming, and are gram-negative rods. The most well-known coliform is Escherichia coli, or E. coli. We test water for coliforms because their presence means fecal contamination, and that suggests other harmful microbes might be there too. Obligate pathogens in Enterobacteriaceae. Some enterics are obligate pathogens, meaning they always cause disease, even in healthy people. These include Salmonella, Shigella, and Yersinia. They are not part of the normal flora and can cause serious illnesses. Why do we study Pseudomonas aeruginosa with enterics? Although Pseudomonas aeruginosa is not a member of Enterobacteriaceae, we often study it together with enterics. Why? Because it's a gram-negative rod, and it causes opportunistic infections in similar clinical settings, like in hospitals, so it behaves similarly in real-world cases, even though it's from a different family. Section 6, Media and Biochemical Tests. In the lab, we use different types of media to identify bacteria. There are 3 key categories. 1, selective media, allows some bacteria to grow, but not others. 2, differential media, shows visible differences like color changes. 3, selective plus differential media, does both if. Let's go over the most important ones. Phenol red broth. This is a differential medium. It's used to test whether a bacterium can ferment a sugar. What's in it? 1 sugar like glucose or sucrose, a pH indicator called phenol red, and a Durham tube that traps gas. Results: Ag, yellow broth plus gas equals fermentation plus gas production. A mass, yellow, no gas equals fermentation, no gas. Red broth equals no fermentation. Just remember, phenol red broth tells you about sugar fermentation, but it doesn't tell you if a bacterium is a coliform. EMB agar. Eosin methylene blue EMB is both selective and differential. Selective. The dyes eosin Y and methylene blue inhibit gram-positive bacteria. Differential. It tells us whether bacteria ferment lactose. Results. Strong lactose fermenters like E. coli produce dark purple or green sheen colonies. Weak fermenters, pink colonies. Non-fermenters, colorless or transparent colonies. EMB is useful to help identify coliforms because coliforms strongly ferment lactose. Simmons citrate agar. This medium tests whether bacteria can use citrate as their only carbon source. It contains citrate, ammonium phosphate for nitrogen, and a pH indicator called bromothymol blue. If the bacteria have the enzyme citrate permease, they can grow and turn the medium blue. Results: blue equals positive, bacteria can use citrate; green is negative, no growth. SIM media, sulfide indole motility. SIM stands for sulfide, indole, and motility. It's a combination test, 3 results in 1 tube. Let's break it down. 1, sulfide. If the bacteria produce hydrogen sulfide, H2S, it reacts with iron in the medium, forming a black precipitate. 2, indole. If the bacteria have tryptophanase, they break down tryptophan into indole. After incubation, we add Kovacs reagent. A red or pink layer means indole is present equals positive. 3, motility. The semi-solid medium allows bacteria to move. If they spread away from the stab line, they are motile. If they stay near the stab line, they are non-motile. Note, if the tube turns black from sulfur, motility might be hard to see. Section summary. Enterobacteriaceae are gram-negative rods that live in the intestines. Coliforms are lactose-fermenting enterics like E. coli. Their presence indicates fecal contamination. Obligate pathogens like Salmonella and Shigella always cause disease. We use media to grow and identify bacteria. Phenol red broth tests sugar fermentation. EMB agar selects gram-negatives and detects lactose fermentation. Simmons citrate tests citrate usage. Sim media detects sulfide, indole, and motility. Review questions. 1. What 4 traits define an enteric bacterium? Gram-negative, rod-shaped, facultative anaerobe, lives in the intestines. 2. What is a coliform, and why do we test water for it? A lactose-fermenting gram-negative rod, it indicates fecal contamination. 3. What result on EMB agar suggests a strong lactose fermenter? Dark purple or green metallic sheen. 4. What does a yellow color in phenol red broth indicate? The sugar was fermented, producing acid. 5. How do you test for indole production in sim media? Add Kovacs reagent after incubation. Red layer means positives. Let me know when you're ready for part 4, where we'll cover microbial control methods, the Kirby-Bauer test, and resistance levels, all explained in the same calm, natural voice format. Awesome! Here's part 4 of your Lab Exam 2 audio lecture, covering control of microbial growth and the Kirby-Bauer antibiotic test. It's written in a clear, natural, and slow-paced narration with a summary and review questions at the end. perfect for screen recording or studying of microbes. This is critical in healthcare, food safety, and public hygiene. We use physical and chemical methods to either kill microbes. Section Awesome! Here's part 4 of your Lab Exam 2 audio lecture, covering control of microbial growth and the Kirby-Bauer antibiotic test. It's written in a clear, natural, and slow. Section 7, summary and review questions at the end, perfect for screen recording.